so grateful to have Matt here with us. But before I talk about all the fabulous things a person could say about Matt, so we are recording. And so if you prefer not to be in any of the recordings, just give us a little message and we can edit you out. And we're so grateful to have so many different folks from different countries, different disciplines, all of this. So please don't hesitate to put any questions in the chat, and then there will be some time for Q&A after Matt's presentation, or he can certainly take any as he goes to if they're relevant to specific slides as we roll through. So that's our getting our housekeeping out of the way. We've been working with Dr. Matt Tegtmeyer for a few years now, and we're like getting to the thick of things. So we're really excited. He's been a tremendous collaborator for us. We have Antoinette on the call, for example, who is doing a wonderful internship with us, also under Matt's tutelage. So we're so grateful to have her with us. And there are so many of his students that have made wonderful contributions to 8P. Basil as well as Autumn, and we're so grateful for his whole lab and the leadership that he brings and his incredible skills at cultivating young scientists. So he is at Purdue now, but prior to that, he was at the Broad as well as Harvard, as well as King's College London. So he's been lots of wonderful, prestigious places. And as of late, what he's been doing is working on cell painting with our 8P cells. So he'll tell us all about it, but there's a lot to come, I think, with our wonderful collaboration with Matt Tegtmeyer. So I'm so grateful that he's here today and looking forward to all that he'll tell us today and in the future. And with my gratitude, Matt, take it away.
Okay, thanks, Lauren. And I'm happy to present and talk about some of the stuff that our lab does, and then also kind of share what we're doing with 8P and now and kind of beyond. And any questions whatsoever along the way, I'm much more prefer kind of general discussions and back and forth and talking at people. So don't hesitate to ask me anything you want. So what I'm going to talk about is image-based profiling and thinking about using image-based profiling, which I'll define here soon, for drug repurposing and kind of disease mechanism discovery in the context of 8P syndrome. So my lab is really interested in kind of a simple but complex question, which is how does genetic variation, meaning DNA mutations, both rare and severe in the context of 8P, but then also just common genetics that each and one of us have that makes each of us different. And how does that affect cells in our body? And ultimately, when cells are affected, tissues are affected, and that gives rise to different types of human clinical phenotypes. Now, the tool that we use to address this question is our induced pluripotent stem cells. And so for those who may or may not be familiar with what an induced pluripotent stem cell is about 15 years ago, some scientists in Japan and in the UK, a fellow named John Gurdon, who actually recently passed away a week or so ago, realized that you could take cells from a living person. So you can take skin cells, you can take hair cells, or even cells that wash off from the bladder and the urine. And you can kind of turn these back into the earliest cells in the human body or something like that, that can then be turned into many other cell types. So we can, from any living person, take a cell and then turn that cell into different models of organs and cell types in the human body that might be affected by different diseases. And so you may or may not be so familiar with, but the Project 8P has done a really great job of ascertaining biosamples from 8P heroes and their family controls, and have generated several different stem cells from these persons. And we utilize some of this in the work that I'll talk about shortly. But this is kind of our approach to understanding human diseases. We want to study cells that have interesting mutations. And we want to study different contexts, so the brain, the heart, the liver, etc. My lab is mainly focused on the brain, but we do dabble a little bit in other systems. And really the reason we want to study cells is because we kind of think at some point, a disease cell can maybe tell us what's wrong with it. So if we just look at a cell and we measure different aspects of it, if that cell has a mutation that we're interested in, so in this case, if that cell has a large structural arrangement on chromosome 8, can changes in that cell tell us about what is going on and maybe what gives rise to more complex phenotypes in these individuals? And so we really like to do this through taking pictures. Now, pictures have for a long time been able to faithfully report on different types of diseases and conditions. Um, it makes sense now if you say look at somebody who has Down syndrome, you can kind of tell they have Down syndrome, right? And computer models and computer vision have done really well at being able to take a picture of an individual and predict a clinical phenotype. So there's, of course, Down syndrome, there's Angelman syndrome, and many other conditions. That's even now applied to things like autism spectrum disorders. But you can also think in the context of skin cancer, of using images in histopathology, or probably one of the most famous examples is sickle cell anemia, right? Where you can look at the blood cells and tell that some cells have a sickle-like shape, right? It's kind of a classical example of just looking at something and seeing that there's something different. And if you think historically, microscopes or looking at images was kind of the first step in experimental life sciences and biology and just trying to understand the world. The first real lens-based microscope was developed in the 1500s by a few Dutch optometrists. Um, and there's kind of this really seminal book in the late 1600s by Robert Hooke, which really applied micrography at the time to all different types of things in life sciences. So looking at different kinds of bugs and frogs and all these different types of things under a microscope to begin to understand kind of their structure and function. And, microscopy and using images has been used since, for the last 500 years to really measure different phenotypes, right? And a phenotype is kind of when we look at something, we can measure an aspect about it and tell it apart from something else. And this is, historically, we've been looking at measuring simple phenotypes. So is just the shape different? Is there more or less of something? And so on and so forth. Or when we think about kind of cancer diagnostics and radiology, can we scan the body and find things that are present that maybe shouldn't be present? And this is kind of, the classical progress of using microscope images and looking at how things appear. But there's been kind of an update and a revolution to this in the era of artificial intelligence and machine learning, where we can actually move beyond just simple phenotypes. And what we can do is we can extract many, many thousands of different characteristics about a given image, such that we can move forward to capturing really complex phenotypes. So when we think about image-based screening, we're trying to measure simple phenotypes. So is there more or less? Is it larger or smaller? But in modern days, where we have machine learning and AI, what we can actually do is create what we call image-based profiles, where it's an amalgamation of many thousands of different characteristics about a cell that we can extract and learn information from. And using this, we can really measure complex phenotypes. So it's not just, is it bigger, is it smaller? It could be, what is the location of different compartments and different features within a cell? And then can we extract enough information to apply more complicated analysis approaches in order to find out new information about a disease or a gene mutation, and ultimately find medicines that can help to restore or prevent the onset of a condition? So when we think about image-based profiling, what we'd like to do is take different conditions or perturbations or scenarios, extract many thousands of characteristics about how that cells from that given condition look, and then try to measure and match similarities between profiles. So we can ask how different is, say, an 8P cell from a non-8P control cell, or to what extent does a certain medication make the 8P cell look more like a control cell. And so we can then begin to either predict what are the morphological features that are impacted by a mutation of interest, and ultimately what medicines might be useful or chemicals might be useful for making those cells look more like the kind of unaltered state. So the most kind of famous tool for doing image-based profiling is known as the cell painting assay. This was invented by Ann Carpenter at the Broad Institute, been now 10 years. And what this is rather than just looking under a microscope and taking one image and measuring one small characteristic about the cell, we actually take many images from the same kind of view, focal point. But within that image, what we've done is we've labeled different types of features of the cell and different organelles. Okay, and so your cells have many different organelles that serve different functions. Okay, so we can stain the nucleus, which is kind of up here in the top left, the endoplasmic reticulum, which shows role in producing proteins and so on. When we use this assay, from one focal point, we extract 5 or 6 different images. And let me know if I freeze because I got the little your internet's unstable thing. But and then what we can actually do from that is we measure many features about each of these organelles, but then we also look at their relationships to one another. And so what you can do is from this set of images, you can actually extract many thousands of characteristics about the size, shape, intensity, texture, and localization across all of these organelles and the different compartments. So what about within the nucleus or just within the cell or the cytoplasm, which is kind of the cell area minus the nucleus? And then what we do is we measure similarities and connections to find drugs and genes or to define disease states and predict the outcomes of certain experiments using kind of more advanced computational methods. Another thing that's really valuable about using image-based profiling from a standpoint of wanting to find new medicines or find old medicines that might work in your case is that there are a lot of public resources using cell painting. Okay, so the most prominent one is called JUMP, which is the Joint Undertaking for Morphological Profiling. And here there were dozens actually of pharmaceutical partners and other sort of types of companies like Google, for example, and AWS and Amazon Web. They kind of got together and created this really, really large data set on almost 2 billion cells, where they've used cell painting on cells that were exposed to about 150,000 different small molecule compounds developed by these pharmaceutical companies, as well as many different.
Did Matt freeze for other folks as well?
Did I freeze?
For me, but it could be my internet as well.
It
All right.
froze for me as well. Yeah.
Okay, I'm going to turn my
Okay.
camera off just in case that is helpful. So,
Great. Thank you.
Thanks, Matt.
yes, and then I'll just restart this slide just in case. So I actually can't even figure out. There it is. Okay. So one of the things that I think makes this valuable is these large public data sets that anyone can use. These are free and open to the public. There's, of course, a barrier to learning how to use them. But what we want to be able to do is measure phenotypes in a disease or a context of interest. So maybe that is AP. And then we want to look and see, of these 150,000 small molecules, which of them might have the ability to restore a given phenotype of interest that we measure? And then is that something worth testing in our cellular models, ultimately to nominate that a drug might be beneficial for one of our AP heroes? So the main question that I think about image-based profiling for is what medicines might make a cell that has a disease phenotype look more like a healthy tissue or cell. And, you know, in this kind of partnership with AP and Lauren and Bina and Katie and everybody, we've really been interested in trying to understand how AP mutations affect brain cells. And so one of the things that I developed several years ago when I was working with Ann Carpenter and others at the Broad Institute was to take cell painting and then use that for neuroscience specific applications. And so we developed what we called neuropainting. And what this was essentially was the cell painting assay, this multi-stain dye assay, but applied to different cell types for neuroscience. So these are stem cell derived brain cells for which we were able to utilize the cell painting assay and develop analytical and image analysis pipelines to measure many different features from these different cells. And initially, we applied this to another neurodevelopmental associated copy number variant. This is the 22q11 deletion. So this is a 3 megabase deletion on chromosome 22, and it's the most common CNV in the population. And now, I think there's...
Matt,
Yes?
I'm sorry. When you say it's the most common in the population, what population?
The
Everybody?
human population.
Okay, awesome.
Yeah.
Thank you.
Yeah, this is 1 in every 3,000 people. So the frequency in the population is the highest in terms of copy number variants.
Do you know what what data set that they used to figure that out?
So that's been kind of a long-standing thing for like 20 years, but that might still hold up in like ClinVar.
Okay, awesome.
Yeah.
Thank you.
Yeah. And so what we first wanted to do was to figure out, well, could we use neuropainting to actually learn about mechanisms associated with this mutation? And so when I was back at the Broad Institute, we had a large collection of stem cells from patients with this 22q11 deletion. And we differentiated those into different neural cell types. So a neuronal precursor, progenitor cell, excitatory neurons, and then astrocytes, which is kind of an immune cell in the brain. We did the neural painting, and what we observed is that when we just take these, thousands of morphology features, we can clearly distinguish deletion from control cells. And I'll just highlight a few examples of kind of how we would then use these image-based profiling tools to learn about 22q11 biology before talking about how we're now applying it in AP. So we can measure differential features, so to speak. So what we ask is, of all of our many morphological features, which ones are seemingly affected by the presence of the deletion? Okay, and in this case, we find many hundreds of these features that are affected by the 22q11 deletion, but we also observe there's a lot of cell type specificity. Okay, so only a small proportion of the features affected overlap. Many of them actually only affect a specific cell type, or maybe affect just a few cell types. And I think this is really important to think about when in a lot of times when we're performing cellular studies, we kind of have a singular cell type of interest that we want to understand. But no human disease is, with the exception of very few, isolated to a singular cell type. And so when we think about whether it's drug discovery or disease mechanism, I think it's really important that we take that into context that many cells are going to be affected, especially when we're talking about large copy number variants. Yeah, Lauren, do you have a question? Is that right?
Sorry, yeah, thank you. I was just wondering, I might have missed how many individuals are represented by each one of the cell type blocks, and if you expect that it's highly variable across individuals, or if you're thinking that there's consistency.
So in this heat map, there's just one cell type shown. But in terms of each of the cell types, we have 24 controls and 24 22q samples. I don't know if that's your question.
I think I was thinking specifically in the Venn diagrams. Are you the differentially expressed genes, are those specific to an individual, or that's when you did the overlap?
Oh no, this is accounting for the differences in people. So this is
okay.
Yeah. So there's no within individual comparison here. We're calculating, all 24 individuals with the deletion, all 24 without, and then asking about the features different between those two groups.
Perfect. Thank you.
Yeah. Yeah. And we can get into things like power calculations and such, but and just as one example before I kind of move on to what I think people here are more interested in, is an example of what we can learn from image-based profiling. So here's an example of a morphology trait. Okay. Now the trait is called cytoplasm correlation RWC ER MITO. Let me explain what this means. Okay. So what we're interested in terms of a cellular compartment is the cytoplasm. All right. So kind of this blanket area here. where here's, can you see my pointer actually?
Yes.
Okay, I just want to. So here's our nuclei, right? Here's our cell outer area, okay? And what we're actually interested in is that outer area minus the nuclei. So it's all of this stuff, right? So we're looking at the correlation between the endoplasmic reticulum and the mitochondria, okay? And rearranged correlation just kind of is a way to say intensity, all right? So what we're actually comparing here is the correlation between the endoplasmic reticulum and mitochondria. So for every place you see an endoplasmic reticulum, is there also a mitochondria? And the extent to which this is true, let's say in control cells, there's a 90% overlap. When you have a mutation on chromosome 22q, that correlation drops to say 40%. Okay. So there's kind of this decoupling of mitochondria in ER. And mitochondria provide energy to different organelles within a cell. And so here what we might think as the ER's primary job is to translate RNAs into proteins, we might think that there's actually this deficit in the ability to make proteins because there's less mitochondria providing energy to that organelle. Okay. And so that's kind of one example of how we might think about what these sort of features may mean and represent. Okay, so now I'm going to talk about what we're doing here with AP and our plans, and then I'm going to give some preliminary results. But I want to kind of shout out a few folks that are working on this. So Basil and Antoinette are both, I think, in this meeting somewhere. They're both graduate students in my lab here at Purdue and are working on different aspects with AP. Antoinette's also an intern, but then Basil and Antoinette are also working on some image-based profiling questions that we're trying to answer. And Autumn is kind of like my lab manager, keeping everything together. And so she's contributed in various ways to kind of all of these projects. So the first question we want to do is similar to what we had applied in the 22q11 deletion, was to simply ask about morphological features impacted by the chromosome 8p mutation in different types of brain cells. And so we've been utilizing the stem cells that were generated from Project AP, from AP heroes, as well as the parental and sibling controls. And we're differentiating these into different cell types. So first we generate these into a neural progenitor cell. So this is a precursor cell. You can kind of think early fetal brain development. Then cells mature and divide and they become different cell types. So in our case, we're going to also generate excitatory neurons as well as astrocytes and other cell types as kind of this project advances and we get new brain models that we're playing around with, we may add things. But then, and Basil is leading this, and then what we do is we do neural painting. Okay, and we measure all 4 of these cell types. So we're not just doing one. We want to understand how the AP mutation is impacting all of these different models. Okay. And what we ultimately want to do at the end of the day is say, what are the morphology features that distinguish AP heroes from their parental or sibling controls in each of these different cell types? Okay, such that we can learn, similar to the 22q, is this, are there relationships between organelles within the cell that might be different, and can that explain to some extent some of the differences we see in the heroes? The other question that's kind of baked into this is there's different types of mutations on chromosome 8p, right? And do these different mutations affect the cells and affect biology in the same way or in different ways? Such that when we ultimately look at new medicines or ways to help the heroes, do we need to think about different approaches based on the structure of the genome here? Okay. And I think the answer will be to some extent yes. So we'll talk a little bit about that. And the other thing that I mentioned briefly and that I think is really critical here is, can we identify medicines that are already approved that might rescue our morphology signatures for AP? And let me walk through kind of how this works. Okay, so we want to do cell painting and measure phenotypes, right, from cells from our heroes and their controls. And when we measure them, we might, find some way in which they're different from controls, right? So inversion, duplication, deletions may differ in one way, deletions may differ in another. They also might differ in very similar ways, but we, are able to measure some sort of similarity between the 8P phenotypes and the control phenotypes. What we then want to do is go back to these large public data sets. And one, for example, would be that large JUMP consortium data that has about 150,000 different compounds. And what we would actually want to do is do some computational analysis, what we call in silico analyses, that would predict compounds that would kind of take our 8P cells and shove them back down more closely to the control cells. Such that can we kind of make the 8P cells more similar in their morphology to the control cells, and see if this is a way to nominate different medicines that are already FDA approved to then test on the cells and validate whether or not they actually do kind of meet our prediction. And then can we think about ways to engage with patients and parents and clinicians that these might be useful for trying, ultimately. And again, here we're talking about compounds that are already approved by the FDA. Okay, this isn't, the 20-year route of new drug development, right? These are things that are already working and being repurposed. And I know 8P has done a lot of work to help to try to identify many of these. But this is just a different approach. We're trying to take a little bit more of a quantitative method to kind of get in at this. So the other aspect that Antoinette is kind of leading, and this is something that has more or less been racking my brain for years. So while I'm excited to apply it in 8P, I think this has been a long-standing question. And one of them is kind of, how do we know what the best cell type to study is? We're in a world with limited resources and limited time, right? We'd love to study all of them and everything, but knowing which one gives you kind of the most bang for your buck is difficult. And this isn't an 8P specific problem. This is a human disease problem. Now, how can we think about what is actually the most effective and efficient way to identify new therapies? And the other question is, do we have to use brain cells for studying brain conditions, right? And neurodevelopmental disorders. Now, people have gone back and forth for this, and I've been more or less, of the mindset, not only because like for my own academic survival, I have to say we have to use brain cells because that's what my whole lab does. But, a lot of these cell painting data sets and things have been done and kind of cells that aren't really that relevant for human diseases. They're kind of in easy to use cell models. Some tumor cells, which of course are useful, but they're and HeLa cells. But at the end of the day, I'm kind of like, how do those phenotypic profiles match to, say, a brain condition? But there's more and more data now that actually suggest different cell types are useful enough to capture signatures for drug repurposing. So one example is a recent paper actually led by Ann Carpenter and Bruce Cohen, who's a psychiatrist, when they actually showed that they could simply use skin cells. These are pictures of skin fibroblasts, so from a dermal punch, to find disease signatures for bipolar disorder, which is an extremely complex psychiatric condition. And in fact, they can use this to nominate new drugs that to maybe try in these individuals. And in their case, they're talking about a mitochondrial phenotype. And so, here's some growing evidence that even if you're studying, let's say, a really complex brain phenotype, a psychiatric phenotype, other simpler to use and more efficient to access cell types might be able to do enough in order to do repurposing. I don't think you're going to learn a whole lot about, say, brain biology here. But if we're in a landscape where what we're trying to do is just use computational methods, whether it's machine learning or artificial intelligence, to find signatures and recommend new compounds, we might be able to do well just looking at, say, skin cells. And so Antoinette is testing this hypothesis. So one of the things that she's working on now is trying to compare the efficacy of what we call somatic cells, so cells we can take from a living person, so say skin cells, for drug repurposing. And so the way she's going to go about this is we, thankfully, thanks to Lauren and Katie, we have many different, not only stem cells, but we also have skin cells from some of these 8P heroes and their controls. And so what we're going to do is test a few different cellular models and ask which ones are we able to most reliably distinguish between 8P and controls, but then also which one, based on these large reference drug repurposing data sets, gives us the best opportunity to predict with prediction accuracy which compounds might rescue that. Okay, so really the critical thing here would be in a landscape where we might need increasingly large numbers of samples. So when we think about how many samples do we need in order to reliably have a phenotype, that might be several dozen, it could be several hundred. And doing that with, say, stem cells is really expensive and difficult. But getting skin cells from a few hundred people is not that big of a lift, right? And so if we get to a point to where for large repurposing, using kind of large computational methods, that skin cells work best, that's going to be the cheapest, fastest route to getting new medicines to our individuals. So she's kind of testing this question now. But I think, and this is something that's broadly amenable to a lot of different diseases. Okay, so I want to talk a little bit about some preliminary results that we have from using cell painting on these cells with AP and some controls. So we started out just kind of testing whether image-based profiling would allow us to kind of measure differences in phenotypes. Because while we definitely would make the assumption that we'd find a lot of morphology differences between the hero cells and then the control cells, you can't kind of take that for granted. We need to actually find out. And so what we did is we started with 3 cell lines, one parental control, one cell line with a deletion on AP, and one with an inversion duplication deletion on AP. We generated neuronal progenitor cells, and then we did cell painting. And this was worked on by both Basile and Antoinette. And so here were just some representative images of some of the different things that we're staining for. So we measured many different hundreds of morphology features, similar to what I talked about with the 22q samples. And what we found was that there were a lot of different morphology features that were different and affected by the mutation. Okay, and so here in the blue line, what we see is there are about 248 features that are altered in the AP deletion samples when compared to controls. And in inversion duplication deletion, we identified 175 features that are affected compared to the controls. And again, similar to 22q, we can look at overlap. So there are most of the features that are affected are shared between the deletion and the inversion duplication deletion, but there still are a large number of features that are unique to each. Okay. And again, I think sometimes when we're talking about like morphology, we're not talking about genes here. So sometimes you have to kind of reorient yourself, right? There are several hundred genes in the AP region. And so if we're doing differential gene expression, we might have a set number we would expect to be different. Here we're talking about different types of things. So a lot of things contribute to morphology being different. So this is always important to iterate that because looking at morphology features is more unusual. Okay. Let's see. Yes. Okay.
Matt, do you see
I'm now looking at the questions because I just saw them pop up.
Awesome.
Let me go back to your question, Lauren, if I didn't already answer this.
I think you kind of got it, but I'm glad that I'm glad I anticipated it. That was a beautiful walkthrough.
We are testing your hypothesis. Okay. Okay, so to Bina and iPhone's question and Faye's question. So I think, and this is partially why I kind of mentioned like these aren't genes thing. And again, this is an N of 1 in each condition. So I don't want to get to, this is our conclusion. There could be many things that affect why we'd have more in one than another. And again, we haven't then gone back to say it's this hero, these are the genes that are up and down based on the breakpoints. But it could be the case if we're thinking about the actual influence that overexpression of a gene could be more or less impactful than underexpression. And there could be, I could try to explain this away with biology, which is that mechanisms to reduce overactive transcripts, I think, are a little bit more efficient than mechanisms to recover reduced transcripts. So for example, nonsense-mediated decay is a process by which RNAs are degraded when they're either in excess or something's wrong with them. And so cells may Yeah, well, I mean, we do know that. I just can't remember in these two folks here.
I imagine you know it. I just we the audience are in suspense about it. Do you remember like which one just like or like how they compare the size of the rearrangements for these two individual heroes?
I don't off the top of my head. I only know they're like Purdue label, but we could dig into that.
Let's
Um,
send that around and follow up. I feel like that'd be
and,
very
and share
interesting.
that. Yeah. So, I like I said, I'm really looking forward to when we expand this to we have about I think, you know, 11 hero cell lines. So this again is just kind of two. Um, but it'd be curious to see if this kind of holds true. Right. And significant here we're talking about by a p-value. And so, slight adjustments in the p-value might bring these closer together or so on and so forth. Um, but once we have kind of the expanded cohort, if we really do see this observation, there's a lot of different things we can look to pinpoint. Maybe are there certain, genes where um, the overexpression of that same gene has very little biological effect, but it's always deleted in the deletion, and therefore there's a much stronger kind of effect. So that will be a great question we can answer for you in a month or so, Lauren, just because with one sample, it's really hard to estimate that. My optimistic hope is that with the 10 or so that we have, or 11, we will be well powered because the mutation is so much larger than the 22q mutation. I'm really hoping that that's going to be the case, and especially
Is
Antoinette's.
it
Yeah.
oh sorry, is the 22q, I maybe missed this, but are they variable? Like they are, right? But maybe not quite as variable as what we see in 8p.
So yes, they're more variable, and it would depend a little bit on how we define variable in this case. So there are four different types of mutations with the 22q deletion. Um, and so it's kind of like where those breakpoints are kind of always the same. There's four sets of low copy repeats on that chromosome. About 90% of the patients have the canonical 3 megabase. There's a 1.5 megabase, and there's a few really small deletions that are kind of proximal. And with AP, it seems to be the case that there's a lot of breakpoint variability, right, due to broad genetic instability there. And so there is much more variability. But I think because it's so much larger and there's so many genes within the 8p, looking at the individual genes and the breakpoints is super interesting. But I think for a broad phenotype, where we're going to be well powered at a lower sample size for that reason. And then if it gets to things like when we tack on to gene expression, for example, and we want to look at whether there are specific genes where those breakpoints, say, cut apart, then we want to kind of get a little more specific. Okay, so I just wanted to highlight basically three features that came out of this analysis. One feature that's only in deletions and not the indel dupes, and vice versa. And then I want to talk about one feature that's kind of shared. And again, these are kind of real preliminary, but I want to give some examples of what we're seeing so far. So in one case, what we're looking at is a morphology feature that's affected only in the case of the deletion. Okay, and this morphology feature is the intensity of the Golgi apparatus. All right. And so what we observe is in deletions only, but not in inversion duplications, there is a kind of reduction of the number of Golgi in the cells. Okay, so the intensity of the Golgi is reduced. And the Golgi is responsible for modifying and packaging proteins and lipids and shuttling them around the cells. Okay. So there's kind of one example of a morphology feature that is affected. Now, again, there's a lot of biology that follows this, right? So this isn't, we're done, right? We want to confirm that Golgi's down. And of course, we want to see that this is true in our expanded cohort. But when we look also at kind of things that are in the inversion duplication deletion samples, what we actually see is a shift in the distribution of the endoplasmic reticulum. So in control and deletion cells, the ER is maybe more equitably distributed around the outside of the cell. But actually in the presence of the inversion duplication deletion, the ER kind of shuttle in towards the center of the cell. Okay. And the ER is responsible for synthesizing lipids and proteins, right? It then gives them to the Golgi. But here, what we're thinking about is, the production of proteins and lipids maybe being altered or just changed in some way in the inversion duplication samples, but not necessarily, at least statistically, in the deletion samples. And then when we move to kind of what are morphology traits that are affected by both, or affected in both deletion and inversion duplication, and one is actually just how much mitochondria there is. And so what we do see is there's a reduced abundance of mitochondria kind of near the outside of the cells in the inversion, duplication, deletion, and the deletion samples when compared to the controls. And so, and this may have been a suspected phenotype, and I know we talked to Jen Muell's group a few weeks ago, but preliminarily we do see that there's kind of this decrease in mitochondria in the 8P samples, at least in the neuroprogenitor cells. So these are just three kind of snapshots that we're expanding to more cells, of course, and more cell types and more cell lines. But at least what we are seeing is that the morphology screening is able to help us to uncover morphology traits that are different and affected by the 8P mutation, at least in neuroprogenitor cells. There's no reason to believe this won't be true in the other cell types. And we're kind of moving forward with expanding to more samples and cell types, and ultimately trying to use this with kind of novel computational approaches to predict medicines that would rescue our cells. And just as a brief example, while it's much more complicated than this, what we'd really be trying to ask is, if we put drug X on these cells, does the value for this feature become more in line with this feature? But we do this when we're looking at all thousands of features, so you kind of create this similarity index, right? And if we push these cells back to control-like state, is that something like a rescue of their function, which we would additionally test further. So I want to, thank all the folks in my group, as well as Project 8P. We've been working together for a little over a year now, and it's really been exciting. And I'll also mention the newest member of our lab and the Project 8P team, which is Asher. She is 6 weeks old today, and has been keeping me very busy. So happy to answer any more questions about anything, and just have a discussion if anybody is not bored. So thank you, guys.
Thank you so much, Matt, for that incredible presentation. I am lucky enough to ask you questions all the time, so please, anyone that has any questions, go ahead and unmute or put them in the chat.
I've got a question. Yeah, claps and congratulations, Matt. That's great news, that baby. So yeah, I think my first question is about like function. So looking at the mitochondrial figure you just showed there, when I'm presuming, but obviously that's always dangerous, that when you're cell painting for the mitochondria, you can't actually measure function, because that's what you're right is what are you, because you're not staining, are you? You're just literally picking up mitochondria through some sort of phase contrast. Is that right? Maybe if you tell us
we are staining
about the cell. You are staining. Okay, so they're fixed cells.
Yeah, so sorry, I did quickly brush over that. So the cell painting assay itself are cellular stains.
Gotcha. Okay.
And so the mitochondria is measured from a live stain. So you put that on the cells when they're alive, and they uptake the mitotrackers, the stain. Okay. The other stains are applied on fixed cells. So we are measuring the actual mitochondria.
Yeah.
Now that doesn't answer function. That's just morphology. So the thing here now, if we recapitulate this observation, okay, and we had a conversation with Jennifer Muell, who's at Rutgers, and they're kind of experts in seahorse and mitochondrial function. So one thing we would like to do is talk to them and say, hey, we see this, and you test this, right? So this is kind of a discovery tool to just say, what do we think might be changing? And then we can go look at the mitochondria through a more validated functional assay, whether it's seahorse or some other antibody approach that might work. So yeah.
Great. Thank you. Yeah, it's interesting. And I think it'll be really interesting as well when you get more samples, to see, because obviously N of one is, N of one, right?
Oh, sure. Yeah.
And what else was I going to say? And are you planning to look at different sort of differentiations? These were done on neural progenitor, is that right?
Yeah. Yeah.
Are
Yeah.
you
So
planning to
we're
push the
gonna,
pathways any other way? I'm specifically, as an 8P mom,
yeah,
interested in gut, right?
sure.
And then, I'm sure that that'll be interesting in comparison with brain, gut brain access and all that hoo-ha.
Yeah, I mean, from my standpoint, I'm very open to any and all cell types that we can make reliably. We have a very well-established model for progenitor cells, excitatory neurons, and astrocytes that works 100% of the time, 80% of the time. Um, I think for like whether it's enteric nerves or some sort of other gut like model, if somebody in my group, Antoinette is sitting right there, or Basil are interested in going down that route, I'm all for it. You know, it's just as a starting place. And I
Oh
kind of
yeah.
we do different types of liver models and things like this, and maybe that's interesting as well. But I also try to be careful of like if I generate a bunch of gut cells and find interesting things there, like I don't know enough about the gut.
Mm-hmm.
I think got to find somebody who does, right? And
Mm-hmm.
I love collaborating, but it just it's an added layer of like unknown for me. But
Absolutely.
um, and as I'm
That's part
becoming
of
older,
the
yeah, go ahead, Lauren.
That's part of the beauty of the team science approach. And that's why we're here as a foundation. And one of the many things that we do is we convene the mats. Not that there are any more, mats out there, but there are other extraordinary researchers we all bring together, which is such a joy. But I just wanted to underscore one of the other things that he mentioned about the utility of the images for other researchers. So there's, of course, the like end products that Matt will produce, but then the images themselves can be reanalyzed many times for different goals. And so what he's really producing is an incredible asset for us to accelerate other lines of research as well. So they're going to be really important pieces that will we be able to weave into some of the other work that we do, with weaving in Matt as well. But it's going to be really exciting getting those results for the rest of our heroes, but then being able to leverage those images for all of these other research questions, including the really important collaboration that Matt mentioned, where we are looking at mitochondrial stress performance and doing this in a number of different 8P hero cell types with a Rutgers collaboration as well. And Matt's been really helpful with that. So this
And
is a great tool for more research. Go ahead.
one of the other things that's nice about images, different than other data types, is like the technology for measuring them doesn't really change. When we think about gene expression, like every month there's a new technology for measuring gene expression. And so, like 5 years down the road, if you're trying to compare to your previous data set, there's going to be complexities that mask what's going on. But images don't change. Approaches for analyzing those images do, but people still look at histopathology from 50 years ago, right, for new models. And so when we generate all these images, whether they're hosted by 8P as well, there's like a cell painting gallery that we've put a lot of images up to that, you can go and reanalyze. Maybe there's a new foundation model. There's a company called Recursion Pharmaceuticals. They're entirely built upon cell painting. They actually use bright field images. That's a whole nother story. But their foundation model is public. You can take our 8P images, throw them in there, and see if they've got any compounds to predict, right? Whereas that's really hard to do with like gene expression that's super noisy and changes all the time. So that's another reason why I really like this. It is really like forever data in some way. And this
Hi, Matt.
you know,
Sorry, I don't know if I'm jumping ahead in line, but I'm sorry if there's some noise. I'm at an event. Thank you so much for what you've been doing and you're committing to your dedication, commitment to our community and really understanding this. I think what's most impressive is the scale that I don't think many diseases are studied in this way, with you having like basically all of the samples from as many AP heroes that we have in our biorepository. and growing, and actually committing to working on that cohort. So I know there's a variety in there of different genetic rearrangements. Obviously, no two AP heroes are exactly alike genetically for us, but could you, just tell us in the next 3 months, what do you hope to achieve in the next 6 months? Just so we kind of have a better understanding of the plan in your lab, and maybe some insights that we could share with the community.
Yeah, sure. I think so. There, in the next 3 months, there are kind of two, progress aspects from both of the projects I talked about. The first one would be from the stuff that Basil's working on, is to have disease signatures or morphology features that are different between AP heroes and the age matched controls for, I think, all of the 11 heroes plus the controls that we have for at least 3 cell types. That probably being the stem cells, the NPCs, as well as the excitatory neurons. That's because the way kind of the process works is the cells differentiate linearly, and they all do it at once. Okay, so the way the assay is set up, we screen all of the individuals simultaneously, and that's part of the scale. Okay, so we're not measuring, AP Hero 1 today, AP Hero 2 tomorrow. It's everybody all at once. But there's just a lot of upfront prep work that kind of goes into like getting everything ready to go. So I think in 3 months, we should have those signatures ironed out. Okay. Now, in 6 months, what I really like, and this is a little bit harder because Basil and Antoinette, they're learning the computational stuff, but that's a larger lift for cell bio folks, is the drug repurposing nomination, right? Can we come up with a list of 20 compounds we think move the phenotypes? And now in this case, we're going to do this on all 4 cell types, right? We want to take each of our 4 cell types and get compounds to predict and see how well those overlap. Because of course, if you get to a point to where you have 4 models and those predicted compounds don't overlap, we have another question to solve, which is which ones do we go for conceptually? Not, I mean, practically we could do them all, And the other thing in like 3 months, I think with what Antoinette's working on, I think we really want to know, when we look at the different models that we study in the context of AP, what model has the maximal phenotype, so to speak? Which model are we maximally able to differentiate between AP cells and control cells? And is that going to be the fibroblast, the skin punches that we have, and we have a lot of, from our patients because we then generate cells from? Or is it going to be the kind of stem cell derived models? Because I think ultimately, as Project AP invests more in making cell lines, we want to make sure that that's actually the best thing to do when it comes to like drug repurposing, right? And so I think like I'd love to have, a 75% confidence to say, look, when we measure these as by cell painting, it is the NPCs that give us the largest mutation signature, so to speak, or is it the skin cells? And then can we use that information to recruit for new biosamples? Or how do we think about like funding? If it's going to be $7,500 to make iPSCs, but it's $400 to get skin cells, well, where's the money most effective, right? And so that's kind of, I don't know if I gave a great timeline to that, but that's how I think about it, right? To at least have some sense of like, we're seeing divergence here between these models, and this is important divergence that we need to contextualize. And we have a bunch of other compounds that Lauren and I have been talking about that are getting tested in the short term. Like in the next month or so, we're going to begin to test compounds that were predicted by other things that AP's done, as well as like clinician discussions. And so I didn't totally highlight that here because it's not part of like the computational repurposing, but we are also looking at that approach as well to kind of get a leg up of not waiting. I don't know if that answers most of what you're asking, Bina, but I'm happy to.
mean, the follow up to that, I'm trying to be careful to not like have this go into like a deep, dark black hole of a conversation. But you know, how do you know when the vision is there's a repurposed, safe, effective drug that moves some measurement, for the better, right? So I'm just trying to be really like lay in how I'm explaining the question. How will we, like, has this been done before is really my question. In a neurodevelopmental disease, has this been done before in a pediatric disease? I know we have adults as well that are affected, but those are kind of the big picture questions. I don't expect you to have an answer. I just kind of wanted to like say it for the audience.
No, I think that's a great question, and I don't know if there is. There are a few cases where companies have made a note that they have been able to, treat an N of 1 or something like this with a medicine that was predicted by some sort of AI model. Now, in the context of like this cell painting approach to repurposing, I don't believe it's been done for any cases of rare neurodevelopmental disorders. Recursion Pharmaceuticals, which is built on this technology, does have drugs in the pipeline for different complex disorders and a few rare genetic disorders, but they're not kind of pediatric neurodevelopmental disorders. So there is a path to doing it, but I don't know that anybody's done it. But that gives us the opportunity to see if it's possible, right? And so that does make it a little bit hard where there's not such a clear roadmap, so to speak, outside of once we get the compounds, let's test a few. Okay, maybe they do something cool and interesting to the cells. But I think that's where like the 8P network can be very valuable, because with MSAB and others and the connections you guys have of like, okay, this is promising. How do we go to, some of the clinicians that are at some 8P clinics and so on, and how do we think about going forward with that, right? And so on and so forth. So I think 8P is a great use case for this. I'm very optimistic that we can learn something. But there's a reason why so much of the pharmaceutical industry has bought into this. They do seem to be very invested financially and intellectually in like this approach for drug discovery. It's just unfortunately the pharmaceutical doesn't invest in rare diseases often. So I think it's our job to try to fill that gap and do it.
Hi, Matthew. Sorry,
Hey.
thank you for this.
Yeah.
I wanted to ask basically, how would you, because you mentioned that you can identify drugs that are already FDA approved, and how is the process of identifying which drugs to test on the cells?
Yeah, so what we do is we take essentially the morphology profiles, so that amalgamation of thousands of features between the 8P samples and the control samples. And mathematically what we do is we compute something called a Euclidean or cosine correlation value. So we look across all the morphology features and we say, how different or similar are they from the 8P to the controls? And that allows us to calculate some sort of vector, right? That would say, if we changed the morphology profile and to this extent, those 8P cells would look like control cells. All right, taking into account all of those. And then what we do is we go into that large reference data set where they've got the same type of data on many different FDA approved drugs. And then what we ask is, how do any of those drugs push cells along that same vector that we're interested in, that would make the 8P cells look like the controls?
Mm-hmm.
And then what we would do, in practice is say, take the top 10 or 20 of those, just because of financial constraints, and see if those predictions are hold true, right? So then we redo the experiments and see if they've kind of pushed. So that's kind of like basically how we do it in a simplistic way.
And there's some key similarities between Matt's approach as well as Unravel's approach and Heavy and Bio's approach, which are two more folks that have generated drug hits that Matt will be screening, but there are some key strengths to Matt's approach too. So we're really excited that he'll be nominating candidate drugs as well. So we'll be testing candidate drugs from 4 places: the Unravel hits, the Heavy and Bio hits nominated directly by our MSAB, and then Matt's hits. So we're really excited for that.
Thank you. Very exciting for us too as parents. And thank you for the work you're doing.
You're welcome.
Yeah.
And we'll be very, diligent about what drugs we test, right? We don't want, if one of the most predicted compounds is some harsh, cancer, chemotherapy, we're not going to give that to an 8-week kid, right? So, once we get our list, we'll talk with Lauren and all the other folks on the MSAB and whatever, and try to say, okay, which ones are reasonable for the low side effect profile and could be given to young people, right?
Yeah.
So,
and I have to say, that's all the hard work that we have a wonderful intern who's getting ready to go to medical school, Tira, who did that with all of our drug hits from all of the places but Matt. So we have a nice pipeline for going through and being able to winnow that down. So that's really exciting, and we're thrilled to keep rolling with that. I think, are we a little bit over, Katie? I think we might be. Okay.
Yeah.
Awesome. Well, this has just been such a joy, and I would love to have a part 2 of this sometime maybe in the new year. And we're so grateful and just looking forward to all of what the future will bring with your lab, Matt.
Great. I'm excited. I love working with you guys. So thanks, everybody. If you need anything, you can email me. I'm sure Lauren's got she can share my email.